You ready? All right, let's get started. So immune system part 2. Okay, so last Tuesday or on Tuesday, right, we did a lot of talking about the immune system, how there's innate defenses, and then there's these adaptive defenses, right? And we learned a lot about how there is variation within the B cell and T cell receptor that allows the binding to and the targeted attack of two very specific pathogens. So you can create a huge amount of antibody and B and T cell receptor diversity through recombination of the exons of those T and B cell receptor genes, as well as continued affinity maturation after a B cell sort of has been activated, selected. And that then continues through somatic hypermutation to continue to refine that clone of the B cell to make the immune globulins that it produces ever more efficient at binding the antigen that is presented on the pathogen. So today we're going to go further. Okay, that was actually a pretty complicated lecture, and there's also some kind of complex concepts in this lecture, one of which is MHC proteins. Okay, these are two major classes of proteins that are absolutely essential for immune system function. Okay, they are going to be essential for both recognition of your body's own tissues, as well and differentiation of that recognition from your own tissues from outside of the body. Okay. Now we touched on MHC molecules and proteins in the last lecture, so we're going to go a little bit deeper today. So first of all, there's two major classes of MHC proteins. We have class 1, we have class 2, and they serve two different roles. They both are going to display peptides, which are just short chains of amino acids that are produced from proteins that have been broken down. Okay, but they're going to display peptides from different sources. Okay, and they are going to interact with distinct cells in order to activate distinct processes when those cells recognize these peptides. Okay, so the class 1 MHC, this is major histocompatibility complex, that's what MHC stands for. Okay, these are going to be expressed on all cells that have nuclei in your body. That's almost all the cells except for erythrocytes, right? Your red blood cells. Okay. Now, there are in the population, like between all of us, we all have a distinct set of MHC genes that we express, distinct alleles. Okay, so there are 3 genes in the MHC class 1, HLA A, B, and C, I think, and And each one of them, you're going to inherit one allele from your father and one from your mother. Okay, so altogether, you're going to express 6 distinct alleles of these MHC class 1s. Okay, however, across the population, there's huge diversity in the sequence and shape of those MHC proteins. Okay, so the set of which 6 alleles you express is almost like a fingerprint of your own body. It is what you are going to be allowed, what your body sort of can recognize that any nucleated cell in your body is going to be expressing those 6 alleles. Okay, so this is an easy way for the body to recognize itself. Okay, now if you got an organ transplant, okay, from some random person, you get a new kidney, stick it in your body. What's going to happen? Is that kidney going to have the same set of 6 alleles of the MHC class 1s? Probably not. Okay, probably what will happen is that it won't, and then your body will recognize that set of MHC class 1s that's foreign and try to create antibodies against it and try to kill that tissue. Okay, so that is why organ transplantation is very difficult. That is why you have to try to find a donor match for an organ. Someone who's just either randomly, just pretty rare, or because you're actually related to the person, okay, that expresses a similar type, a similar set of alleles of your MHC class 1s between the donor and the recipient. Okay. And only really in the case of identical twins will you get an exact match. So even, but a partial match is still better than no match. Okay, and then you can usually use immune suppressing drugs in order to try to maintain the health of that organ transplanted tissue over the rest of your life. Okay, so that's one thing the MHC class 1s do. They're providing you like a little fingerprint of, hey, I am from my own body's tissues because of that 3 sets of genes. Okay. The second thing that it's going to do is that it is going to also display a fingerprint of everything that is made within that individual cell. Every protein that is produced, and the cell's making many different proteins, right? Every protein that is produced inside a cell eventually gets degraded. It eventually breaks down and needs to be replaced. That process of degradation generally is an active process where it will get tagged with a molecule called ubiquitin, okay? And the more ubiquitins you stick on the end of this little protein, of any protein in your body, that directs that protein to get sent to the proteasome, which is like the little garbage disposal of the cell. The proteasome is going to pull onto that ubiquitin, yank the protein apart, and chop it into pieces, okay? Which are peptides, these short amino acid sequences. Okay? Now, once that occurs, those peptides get sent to the endoplasmic reticulum and get fused onto MHC class 1 molecules that are in the ER, and then those are then transported out and displayed on the surface of the cell. So coating every cell, every nucleated cell in your body, you have these little pedestals of MHC class 1s that are displaying fragments of every protein that you produce. Now, this is great because now it allows cells that are outside of your body, outside of the cell, sort of traveling around, maybe they're in your lymph nodes, okay? They are now able to peer into the inside of the cell with actually not having to penetrate the cell, right? They get to see what's going on inside. And normally in most cells, what's displayed is going to look normal. Okay, you're going to have the appropriate set of proteins, but sometimes in the case in which a cell is infected with a virus or is cancerous, those proteins are going to look unusual. Okay, those peptide fragments are going to look unusual. This is dead. So we'll take it off. The other one has a crappy binding thing. So I'll just keep talking like this if that's okay. Loud enough? Okay. So we have peptide fragments, right, that are displayed. And if you are an infected cell, you're a virus. What does a virus do? A virus typically has an RNA payload that gets into the cell, gets into the cytosol, and then hijacks the protein production machinery of a cell to say, make my proteins, not your proteins. Okay, so that virus gets in, starts making viral proteins. Those viral proteins are going to get also chopped up like every protein that gets made inside the cell, displayed on MHC class 1s. And now we have some really weird peptides on the surface of the cell, virally produced peptides. And that is going to allow a cytotoxic T cell to recognize that as an unusual peptide. I am going to attack this cell. This cell must be infected. Okay. The other way that you can get unusual peptides is if you have a cancer cell. Cancerous cells usually have one of the features of a cancerous cell is that it has undergone mutations that now allow it to replicate sort of endlessly, okay, and rapidly. And it tends to accumulate mutations, and it makes the proteins that are normally being produced maybe have different shapes, okay? So now you could be overproducing some weird peptide fragments from proteins that have undergone mutations that are still your own, they're your own genes, but now because of those mutations, they look weird, okay? Okay. So, and so whatever, yes.
Do the peptide fragments get placed in this like receptor, like class 1 MHC molecule? Like they place there, or they place all across the stomach?
They are placed inside this receptor. And that receptor, they're covalently bound to the receptor displayed on the surface. Okay. And then that T cell receptor is actually going to bind to both the pocket of this class 1 MHC and the antigen fragment itself, and the peptide. Okay. Okay, so if a cytotoxic T cell recognizes this as an unusual peptide, it is going to attack. And I'll talk about how it attacks in a little bit. So that's class 1, all our cells with nuclei. Now, yeah, do you have a question?
Yeah. So is this MHC allele focused more on protein molecules or protein channels, or how is
All proteins. So a molecule is just a molecule is it's 2 atoms or more that are stuck together by covalent bonds. A protein is a string of amino acids that are put together. Okay. And a channel is just some subclass of proteins that makes holes in the membrane, usually via some gate. Okay.
other molecules like saccharides?
Okay, so lipopolysaccharides. You could potentially have those being broken down there. Usually the sugars are attached outside the body. So anyway, but you know, I can continue that discussion after class. So what about class 2 MHC molecules? So class 2 are different, okay? They are not displaying stuff that is produced by the cell itself, okay? And they're not displayed on the surface of most cells. In fact, these are found only on antigen-presenting cells, okay? What are antigen-presenting cells? We have the professional antigen-presenting cells. Those are things like macrophages, dendritic cells. Their entire job is to eat pathogens and microbes, destroy them, and display antigens on their surface, okay? We also have B cells can also eat a particular antigen fragment that it binds onto. Okay, so B cells aren't like going out there necessarily attacking and digesting whatever microbe they can find, but if they do bind to an antigen, they can phagocytose that and display that antigen on the surface. So those are the types of cells, and only those types of cells that display MHC class 2. So MHC class 2, these peptide fragments that they display are produced in a different way. These peptide fragments are from the lysosome. Okay, remember you had some phagocytosis, like on one of the first slides that we had, it gets fused with the lysosome, then basically different enzymes come into that little vacuole, okay, break down the peptide fragments, and those are then going to get bound to class 2 MHC molecules and displayed on the surface. So class 2 does not display what you're making on your own, it displays what you just ate and fragments of what you just ate. And that is class 2 MHC is then going to interact with a distinct set of cells, these T cells, okay, that will then binding of a T cell, in particular a helper T cell here, to this site will now help activate a helper T cell and trigger its proliferation and its ability to then activate other B cells that recognize the same antigen. Okay, so this MHC class 2 is really about coordinating an immune response against a microbe or a pathogen that has invaded you, rather than causing an attack on the specific cell itself. Okay, so MHC class 1, if you get active, if the cytotoxic T cell recognizes something on your MHC class 1, you're in trouble. This cell's going to die. Okay, this cytotoxic T cell will kill it. If a helper T cell recognizes a peptide on MHC class 2, that just activates this and primes it to then sort of go out and find other B cells that like this antigen and activate them to mount an attack against that pathogen. So the MHC class 1 and class 2s, okay, they're both pedestals, right? And they are basically the interactions between either the cytotoxic T cell on MHC class 1 or the helper T cell on MHC class 2. Those are stabilized by an accessory protein that is specifically expressed either by the cytotoxic T cell or the helper T cell. So cytotoxic T cells express CD8. Helper T cells express CD4. Okay, CD8 only binds to MHC class 1. CD4 only binds to MHC class 2. So this sort of side interaction is what is going to allow you have to, in order to have a really long-lasting interaction between the cell displaying the peptide and the sort of target T cell, you want to have a high affinity interaction here where the T cell receptor is binding the peptide in this pocket, okay? And also the CD8 sort of protein here is going to bind onto the side of that MHC class 1 and hold it in place for a while. Okay, so that's going to stabilize that interaction, and that will allow a bidirectional communication here between the cytotoxic T cell and the expressing MHC class 1 or the helper T cell and MHC class 2. So the expression of either CD8 or CD4 is how we get some selectivity over sort of which MHC classes we're binding to. Okay. Yes. Where does the protein fragment go? On this diagram, it's right here in red. So that's the little protein fragment that's displayed. Okay. So what? By the way, so CD, there's all these different CDs. Okay, CD just stands for like cluster diversity molecule. Okay, it's just a way of organizing all the different crap that's on the surface of these immune cells. Okay, they just call it CD8 or CD4. Okay. So let's take a look now at sort of the first, what happens if you get strong binding and recognition of a peptide, and a neutral peptide here, by a cytotoxic T cell, okay, that's displayed on MHC class 1. Okay, what'll happen? Okay, so our cytotoxic T cell is going to have an T cell antigen receptor that recognizes this peptide. Normally, it won't recognize a peptide because it's been screened against any peptides that are normally produced by the body. But now it's found one that it hasn't seen before and that it has a high affinity to. It binds this. Its accessory protein says, okay, this is on MHC class 1, not on class 2. I should try to kill this cell, this affected cell. Okay. So what the cytotoxic T cell will then do is it is going to excrete and drop these granules of different enzymes that are called granzymes and perforins. The perforins will intercalate into the cell membrane and poke a hole in the cell membrane. Okay. Then granzymes are going to come in and trigger, basically they act as transcription factors that will cause the initiation of apoptosis within the cell. So it tells the cell to kill itself. It also could potentially cause the cell to necrose and die through these pores and a rupturing of that cell. Now, how do we actually get that? So this is after the cell dies, the cytotoxic T cell gets released. How do they actually do this? There's a couple different ways. So these are granzymes and perforins. A cytotoxic T cell will actually, once it binds, it starts spreading and smearing out itself all over the surface of the cell. Okay, and it has little actin filaments that it can extend, and it can sort of bind to the cell and then start stretching the cell membrane, the target cell membrane, by extending its actin filaments, stretching out this membrane, and then in the center of the cytotoxic T cell, it drops these granules right onto the surface of the target cell. Okay, that tension from pulling that membrane taut is what allows the perforins to get into it and specifically kill this cell that it's attached to and not the neighboring cells that it might be around. Okay, so it's a very targeted delivery of these cytotoxic killing enzymes only to the cell that it's interacting with. Okay, this is sort of a scanning electron microscope of what it looks like when it's dying. Okay, so that is what happens if you're binding onto MHC class 1. Cytotoxic T cells killing a cell directly. Okay, what about these MHC class 2s? What if you are a helper T cell and you come across a cell that's on MHC class 2 or a peptide on MHC class 2 that you recognize? Well, if it's on MHC class 2, we know that it has to be an antigen presenting cell, something that is phagocytosed to this pathogen, is digested, is displaying its fragments on its surface. Okay, and the first step is that the helper T cell recognizes this antigen displayed on class 2. Okay, the interaction is stabilized by an accessory protein. Okay, so and then there will be a release of cytokines from the antigen presenting cell onto the helper T cell. The helper T cell, if it's continuously in the presence of these cytokines and it's sort of getting stuck onto this antigen presenting cell because of the strength of these two interactions, it will also release other cytokines on itself that can sort of provide positive feedback to itself and to the antigen presenting cell in order to activate the helper T cell. Okay. So once the helper T cell is activated, what it will do is it is going to proliferate. Okay, it's going to make copies of itself, clone copies of itself, okay, which will then travel to lymphoid tissues, okay, and look for B cells that share its same its affinity for a particular antigen and specifically activate those B cells. Okay, it will also produce cytokines that will amp up the role of sort of increase the activity of cytotoxic T cells to say, hey, we are being, infected. Go get more aggressive about attacking other cells that you're scanning for. Yeah.
So like, what's the setup? How does the cell, was that like class 2 MHC just release it all the time to attract the helper T cells? Or
No, only this is a specific interaction when a binding event has occurred and it's been stabilized. Okay. 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 Yes. So a cytokine is something which attracts another cell, something that one cell can release into the molecule or protein that can travel, and the other cell has a receptor for it can recognize that, and it will want to travel towards that location, and potentially it can drive the release of some second messengers inside the cell to change the gene expression profile of the target cell and to transform its functions to basically activate it and go into attack mode, essentially, or to go into helper mode. So it's a general class of molecule. Yeah. So how does the cytotoxic T cell has already been screened in the process of maturation, or it's already looked at many different, before it is allowed to get activated, it's already been screened by looking at many different peptides that the body produces. Okay, and it has been shown to not react to any of those peptides. It's been able to sort of suppress it after that. It doesn't have super high affinity for any peptide that the body normally produces. So then if it comes along and finds a peptide that it can bind to that the body doesn't produce, or if it finds, there's also sort of it also has an ability to find some of the common motifs that are common to many pathogens. If it finds one of those, it also can get activated with those. So it sort of knows like CPG sites, unmethylated CPG sites, those lipopolysaccharides that we talked about in the innate immune system. cytotoxic T cells can recognize those oftentimes. So that does not really require, that interaction does not require helper T cell activation. It's helper T cell independent for these common motifs. And then the more specific motifs, like if you have like a really weird peptide that you've never seen before, that's unusual, that sometimes may require a helper T cell to sort of release cytokines onto a cytotoxic T cell to tell it to attack in that. Okay. Yeah. Great question. Let's look at the next slide. How do helper T cells? Okay, hold on.
Yo, wait, is it like the T cells like make copies of themselves once they're activated? Like,
Yeah, helper T cells will create copies of themselves once activated.
so they don't just recruit other.
No, so you wanna have more than one, so if you have one helper T cell that gets activated, it's still gonna have a hard time finding its pair. Its mate that shares a common affinity for that antigen, right? So you wanna make a lot of copies of yourself. So now you have a chance to find a B cell that is also going to get activated by that pathogen. Because,
Copies of themselves, those like blue dots that are released, the motifs you call it, those is what calls the B cells and X cells.
okay, the blue dots are the cytokines. Okay, a motif is, what I mean by a motif is a common shape of like a protein or a structure that would be common to many kinds of pathogens of a particular class. Okay, so like all the funguses have some like sugar on the outside of them, or most funguses have a sugar on the outside of them that your body doesn't make, but cytotoxic T cell sees that and is like, oh, this must be a fungus. Let's kill it this way, right? With the drosomycin, so we'll make some drosomycin or something, right? Stuff like that. Those are what the folate receptors do. Okay. Yeah.
Cytokines are released. Does it change like the cytotoxic T cell structure to be able to specifically target that one? Most of that the helper T cell found, because it sounds like cytotoxic T cells can bind to many different foreign protein structures.
Mhm.
Does the cytokine like being released changes only be able to target that one specific protein?
They don't change the ability of the cytotoxic T cell. They don't share some sort of recognition of that trait directly. They don't say, hey, this is like the shape I found. Okay, that's not transmitted by the cytokine. But because these are usually found in a lymphoid tissue, right? If you have some helper T cell that gets activated in one lymph node, right? That's probably where the pathogens are. Okay, so it locally is going to turn on the cytotoxic T cells there to get more aggressive. Okay.
It's activating in that area.
Yeah, exactly. So that's where you're getting some of that specificity. Okay. I'm going to keep going for a minute. Okay. Hold it in. Okay, so your question was, how does B cell activation work? How do the helper T cells sort of promote B cell activation? Okay, so this is how it works. So here is our antigen presenting cell. Okay. It activates our helper T cell. Okay, so our antigen presenting cell, could be a professional cell like the macrophages or the dendritic cells. Phagocytosed pathogen, it's displayed. It's now activated the helper T cell. We already went through that process. Okay, this helper T cell is activated by this specific antigen it's really good at binding. Now that this helper T cell is activated, it sort of releases itself from the antigen presenting cell. Okay, it's making clonal copies of itself, and it goes around in the lymphoid tissue until it finds a B cell that is presenting an antigen that it specifically binds to. Okay, so a B cell, as I've said, it's not a professional antigen presenting cell. It's not going around phagocytosing everything it finds. Instead, a B cell mostly just goes around and tries to bind onto things and usually doesn't. But if it does bind onto something, okay, that's great. It can then phagocytose just that thing that it binds onto. Okay, and it will display just that antigen that it specifically binds to. So now the B cell is displaying on its MHC class II that antigen fragment, okay? And if an activated helper T cell can bind that same fragment, that is where we get the real activation of the immune system. Okay, now the activated helper T cell is going to be in close proximity to this sort of bound B cell. Okay, it's going to release cytokines on the B cell, and that is going to trigger the proliferation of the B cell, and then the forming of the germinal center and the the making of the memory B cells, the plasma cells. This is where we're going to finally mount a very aggressive immune response. Now you can imagine why we might want this to happen. Okay, why do we want to have a helper T cell and a B cell both have to get activated by the same antigen, then find each other, and then sort of turn on a response? Because if it's just a B cell, we want to have some sort of backup, some safety sort of measures that are happening in our immune system, right? The B cell there's millions of these different B cells, right? Okay, maybe it finds something that maybe one of those B cells didn't, slipped through the maturation process and is attacking your own body now and wants to produce a bunch of antibodies to it. If we didn't have this co-requirement of getting activated by a helper T cell, by an activated helper T cell that recognizes the same thing, we could probably have a lot of B cells that are inappropriately converting to plasma cells and producing antibodies against ourselves. Okay, so this is sort of like a safety feature. You have to have the agreement of a helper T cell and a B cell that both are recognizing the same thing to say, okay, this really is a bad pathogen. We need to mount a response. Any questions about this? Okay, so now we'll sort of look here, and this is the overall summary of what happens and the timing of how this happens. Okay. First, okay, on the left side, we have the humoral immune response. That's the immune response that's happening in the blood and the lymph. Okay. On the right-hand side are cell-mediated immune response. We have a pathogen that comes in. It's expressing an antigen. That antigen is sort of engulfed by an antigen presenting cell, and also comes over here and binds onto a B cell. Okay, that antigen presenting cell, okay, can now get activated, or the antigen presenting cell will find a helper T cell that binds that antigen. Okay, that is going to activate the helper T cell. The helper T cell will promote the activity of local cytotoxic T cells in that area to try to start attacking the local machinery. Also, this helper T cell can come over here and activate a B cell. Okay, so the B cell is going to bind, get partially activated. A helper T cell that's activated will then complete the activation process of the B cell. The B cell can then proliferate into plasma cells, which are antibody factories. Okay, and the memory B cells, which are going to be stored in reserve for a future infection. Okay. The cytotoxic T cells can also sort of have a memory component and an active component. And then finally, these memory helper T cells that are sitting around here, okay, if you've cleared the infection, and then a few weeks later, or a few months later, or a few years later, you're exposed to the same antigen again, now we have a huge pool of these B cells and T cells that can recognize that antigen more rapidly and provide a much greater sort of higher starting point in terms of the number of cells that recognize that and mount a much more effective response much more quickly. Okay, so what about these antibodies? Okay, what are these antibodies even doing? Okay, what do they look like? How do they function? Antibodies or immunoglobulins, okay, are molecules, proteins that do not directly kill a pathogen, but they do mark pathogens for destruction via a variety of means. Okay, so what are those means? So 1, we've got neutralization. 2, we've got opsonization. And 3, we've got activation of the complement system. Okay, so let's take a look here. This is the shape of a antibody. Okay, or immunoglobulin. This is a soluble form of immunoglobulin called IgG. We'll talk about that in a second. This is a virus. This is the electron micrograph of a virus. What do we see on the virus? And these are to scale. Okay, so this is 15 nanometers across. This virus is maybe 100 nanometers across. Okay, you can see it's sort of This is, I believe this is a SARS-CoV-2 COVID virus, okay? You can see its little spike protein sticking out here on its surface, okay? And those spike proteins are designed to recognize a specific receptor in your lungs, the ACE2 receptor, bind onto that receptor and use the energy of that binding interaction to inject its RNA into the host. Okay. Now, if we've got a bunch of antibodies floating around, okay, that also recognize the spike protein, okay, they can bind onto that viral particle at all of these spike particles on the outside. Okay. If you coat the surface of the virus with a dozen antibodies, okay, now all of a sudden you've produced some steric hindrance. You basically put like almost like a shell that prevents this virus from getting close enough to the cells within your body to actually bind onto the receptors that it's targeting. So it's basically blocking the ability of the virus to touch the receptors that the virus is looking for to get into. So this is called the process of neutralization. You can imagine it takes a heck of a lot of antibodies circulating in your system in order for this to be effective, because there's usually not just one viral particle floating around. There could be billions of viral particles floating around during an infection. So we're going to need billions and billions of antibodies. Thankfully, each one of our plasma B cells is pumping out 2,000 of these antibodies every second. So that's the process of neutralization, basically directly blocking something like a virus from getting in. Now, we also have a process called opsonization. So opsonization is going to be a method that is going to promote phagocytosis, or the eating by a macrophage or neutrophil of a particle or a pathogen. So it could be, you could opsonize viral particles, you could opsonize bacteria. And what happens there is you're going to get each of these immunoglobulins, the antibodies, has a Y shape. Why do we even have that? Why do we want two binding sites? Because we could bind to one particle or one bacteria on one end and another bacteria on the other end and stick them together. You can start clumping the viral particles together or the bacteria together. And that makes it easier for something like a macrophage to actually target and phagocytose that. Instead of eating one particle at a time, can eat 10 or 20 because they're in a clump. Okay. That clumping also, the process of binding here, also exposes this common region on the other end of the antibody, the region that doesn't directly interact with the antigen, but it does interact with receptors on the surface of phagocytic cells. And it promotes the phagocytosis. If a phagocytic cell sees one of these sort of ends here, it's going to start extending its pseudopodia and try to endocytose whatever is attached to this. So it's a promoter. Opsonization is the process of clumping together and promoting the internalization of pathogens. Okay, now, the third way that we're going to attack, that immunoglobulins will attack pathogens, is through the activation of the complement system. Okay, so this is best done by one form of, we're going to talk about the 5 forms of immunoglobulins in a little bit, but this is best done by IgM, okay? But it also can be done by IgG. What happens here is that if we have some sort of foreign cell that's in our body, a pathogen, that is displaying an antigen, and we get binding at multiple sites of these immunoglobulins onto the cell. Again, this sort of the butt end here of the antibodies is sticking out, and it can bind to a protein called complement 1. Okay? Complement 1 then can recruit a bunch of other proteins in a cascade that will come together, okay, and then form what's called a membrane attack complex. When all of these proteins sort of cluster together, and they are only going to cluster together in places where you have a high density of these antibodies bound to an object, okay, it will then create a membrane attack complex. This has a set of proteins that enable it to insert themselves into a cell membrane, create a pore, and then open a pore that allows water to flow. Usually the concentration of proteins and solutes inside a cell or inside a foreign cell is much higher than outside. So water will rush in to try to balance the concentration through osmosis. Okay. And that will cause the cell to swell and lyse. Okay, pop it open. Okay, so that's basically the three different ways that antibodies and immunoglobulins can destroy pathogens. None of those ways it's directly doing it, but it is sort of facilitating that process. It's either directly interfering with the ability of the pathogen to cause damage to your body's tissues, or it's recruiting other cells within your body or other molecules within the body to directly attack. So I mentioned that immunoglobulins have a couple different forms. Now, all of the different forms, and these forms can be, they all share a common motif, structural motif. Here where we have a Y-shaped arm, we've got our two heavy chains that are bound together, bound to the light chain. They all are going to share from the same B cell. They're all going to share the same variable region because they have that specificity. The variable region here that comes together is that unique specificity that that particular B cell that got activated has. Okay. However, they also have this sort of common area, and this common area can undergo a process, can get swapped out over the course of maturation of a B cell and proliferation of a B cell, and through the course of an infection can change the class of immunoglobulin that is being produced. So all of regardless of the class, they're all going to have the same affinity for the antigen, okay? But they can have different functions. So the process of changing out this sort of common region is called class switching. Okay. So we have IgD. Okay, so there's going to be 5 classes. IgD, this is something that has a transmembrane domain. It can be displayed on the surface of B cell receptors or B cells that haven't yet been exposed to antigens. Okay, it sort of looks like that B cell receptor. The first immunoglobulin that's produced by B cells after they get activated is called IgM. Okay. In IgM, there's some recombination of the DNA that when the B cell gets activated, that is going to produce a set of these immunoglobulins of Y sort of hands that form a pentamer, okay, that then can get secreted out of the cell and float around soluble into the bloodstream or into the lymph. IgM, it's got now 10 binding sites. You can imagine it's probably really good at stuff like opsonization, because it has 10 different spots they could bind to different pathogens or antigens, right? The common area in IgM is incredibly potent at recruiting the complement system and generating that membrane attack complex. So this is what's sort of released first. Okay, it sort of initiates an attack via the complement system, but it is not usually the final form that B cells are going to be produced. Once a B cell gets turned into a plasma cell, it's going to start producing a different form. Okay, it's going to produce IgG. This is the most common and the highest concentration of the most abundant immunoglobulin that you'll be finding in the bloodstream. Okay, so these start getting produced a few days after infection, after the B cell gets activated. Okay, you turn into a plasma cell and you become an IgG factory. Okay, so you're constantly going to be secreting this into the blood, into the lymph. It can be present in other tissue fluids. It can neutralize, it can opsonize, it can cross-link antigens. It can still activate the complement system, but it's not as good at IgM as IgM. It's small relative to the other ones. So it is the only immunoglobulin that can cross the placenta. So if you have, if you have a fetus, right, that's developing fetus, the developing fetus does not have its own immune system yet. Okay. So when a baby is born, there is no active immune system that's turned on yet in the baby. Okay, so how is it going to protect itself from disease? Okay, well, it is going to share all of the IgGs that were inside the mother at the time of birth are also going to be present within the baby's blood. Okay, so that is going to provide a passive immune, maternal immunity for the first few weeks to months until those IgGs eventually break down. Okay. Now, there's a second way that you can get passive immunity, okay, or maternal immunity, and that is through IgA. Okay, so IgA does not cross the placenta. Okay, instead, it's a dimer like this and can be secreted, and this is found in tears, saliva, within the GI tract. Okay, the gastrointestinal tract. It lines all the mucus areas. Okay, it's excreted in a specialized way to do that. Okay, it's also found in breast milk. So breastfeeding will allow IgA to enter the baby's gut and provide a barrier defense there, sort of first line of defense against pathogens that the mother's been exposed to. Yeah. So, so this is one of the reasons why, in general, the breast milk is a superior form of nutrition for a baby than formula, because often for a number of reasons, but one of them is because it transfers immunity, whereas a formula doesn't have any IgA. Yeah.
Are we gonna need to know, like, are you gonna test, like what's the shape of this, like give us an image, that and be like, which immunoglobulin is it?
You should know the general structure and function of each one of these forms. Okay, great. All right, now, then finally we have IgE. Okay, IgE is found in the blood. It's not as high, it's not as common as it's sort of the least, it has the least abundance out of any of these forms that's circulating through, but it has a specific role in activating basophils and mast cells, which are found in your body's tissues. Okay. And this is what allows mast cells and drives mast cells within those body's tissues to release granules of histamine and cause a localized immune or inflammatory response. So IgE is very important for recruiting a local inflammatory response. Okay. Now, the thing is, as you also can develop allergies though, right? So allergies are sort of an emergence of inappropriate response of IgE to mast cells, inappropriate signaling, where if you're exposed to an allergen over and over again, much like if you're exposed to a vaccine or exposed to a pathogen over and over again, you might get more and more memory cells, right? You might mount a bigger and bigger response. Similarly, with an allergen, if you start to develop a response against that allergen where you have an IgE, which specifically recognizes that allergen, like, I don't know, cat fur, for example, dander, okay, then that can cause an inappropriate response and cause inappropriate release of histamine, both locally at the location of the site or even systemically throughout your whole body, right? So one response to, say, a peanut allergy or something where you have a shellfish allergy, this can be life-threatening, where you have a systemic release of histamine all throughout the body, causes vasodilation throughout your entire body. That's going to cause precipitous drop in blood pressure because now you have much more space for the same amount of blood, and swelling of the face, swelling of the windpipe, areas around the windpipe, and that can basically choke you and asphyxiate you. So the response to that is we're going to inject epinephrine, like an EpiPen, which is a vasoconstrictor of muscular tissues, vasoconstrictor of vessels within most of our body's tissues. Okay, and that will sort of return the body back to normal. Okay. Any questions about those immunoglobulin structures? All right, so we've talked about the sort of going back briefly to the circulatory system, right? The circulatory system is moving oxygen around and carbon dioxide around through red blood cells, right? Now, the red blood cells are non-nucleated cells, bags of hemoglobin essentially, but depending on your genetic makeup, they do potentially express a few antigens, okay? Your blood type is going to be determined by which antigens are expressed on your red blood cells, okay? So, and there's basically 3 classes of antigens, okay? These are sort of look like carbohydrates that are on the surface of some bacteria. We have antigen A, antigen B, and there's also a family of antigens that are closely related called Rho. Okay, so Rh factor is our sort of third component here. Okay. And so your blood type is based on, do your blood cells express antigen A, antigen B, antigen A and B together, or none of the above? Okay. So this would be type A blood, type B blood, type AB blood, type O blood, and then if you express Rh, then you would be Rh plus or Rh minus. So now the complement of this, so on your red blood cells, if you have type A blood, then your blood plasma, okay, will not express any antigens again, or will not express antibodies against antigen A because that's part of your cell, right? That is your B cells have already been exposed to antigen A during the process of maturation. It's not going to make antigen A. It's not going to make antibodies against antigen A. Okay. However, because antigen B looks very similar to these sort of sugars on the surface of bacteria, you will tend to make an antibody against something that is reactive to antigen B. Likewise, if you have antigen B on your erythrocytes, you will not make any antibodies to antigen B, but you will make anti-A antibodies. If you have A and B, then you're not going to make antibodies against either. If you have type O, then you're going to make antibodies to both A and B. Okay. Yeah. They have a similar shape to the bacteria. The sugar coating on some bacteria really looks similar to antigen A or antigen B. Mm-hmm. Mm-hmm. Yeah, so that's a good question. Is there some like disadvantage if you have type A blood? Are you then more sensitive, more vulnerable to bacteria that have that type A shape? Probably, but a bacteria has more than one antigen on it, right? More than one epitope. So you're probably going to be able to recognize that in a myriad of other ways. Right. So if we have type O negative blood, that's where we don't have antigen A, antigen B, or Rh factor. Okay, we can give those red blood cells to anybody because they are not going to provoke an immune response in any recipient. If you have type B blood and you may have anti-A antibodies, you cannot accept any blood that has antigen A on it. Okay, you can accept type B blood. Okay, you can accept type O blood, but you cannot accept type A blood or type AB blood. Okay. Yeah. Usually they spin it down and there is that's a good question. Okay. But you can basically condense the red blood cells and you want to deliver the red blood cells in the transplant, in the donor rather than just the plasma. They separate out the plasma. Okay, so the universal blood donor is going to be type O negative. You don't have anything on the red blood cells. The universal acceptor, okay, the person that can take any kind of blood is type AB plus, because your blood has all the different antigens that are on it, and your plasma and your body is not making any antibodies against any of those things. Okay, so this is why only about 7% of people have type O negative blood. They are the most sought after blood donors because they can be given to anyone. Okay, any questions on that? All right, so now let's look at viruses, okay, and antigenic variation. So there's a lot of different viruses out there that are pathogenic to humans. They're not all pathogenic to humans, though. Actually, most viruses are not pathogenic to humans. You've got bird flu, you've got swine flu, you've got pigs. I guess that's pig flu, swine flu are the same thing, right? So here's a structure of influenza. Okay, it's sort of packed with 8 different RNA strands here that are going to each produce a protein that is required for the replication and propagation of the virus. Okay, now influenza, depending on the surface of this coat proteins and the composition of these coat proteins, it can either target bird cells, or it can target pig cells, or can target human cells and be infectious to different species. Okay. Now, every year, there's millions at any given moment, there's millions of birds or more that are infected with flu at some amount of time, right? And a bird flu. And there's humans that are infected with influenza, right? And these RNA sequences are constantly mutating. Okay, so every year there's a whole new set of sort of variations in the surface proteins that basically evolve from the sequence of these RNA sequences. Okay, this is because over time, if we're exposed to one shape of this coat protein, right, we will develop antibodies against that and we'll be able to fight it off, right? If there's a new variation, though, that evades our immune response, then we can be vulnerable to that and infected. So the virus is constantly sort of getting blocked by the population as the population sort of evolves its immune response, adaptive immune response against it, okay? And then it finds a way around by changing the composition or changing the shape of these proteins to sort of move its way around antibodies that have already been produced. Now, every decade or two, something bad happens, which is where a very bad bird strain will infect the same sort of creature as a very bad human strain, okay? And these sort of viral genomes, since there's 8 sort of strands here, they can reassort and combine some of the strands from the pathogenic bird strain and the human strain, repackage them into a strain that can infect both birds and humans, okay, and can be deadly. Okay, so the sort of worst influenza outbreak, and really the last real big pandemic before SARS-CoV-2, COVID-19, was the 1918 outbreak of influenza. Okay. And that was derived from a mixing of bird and human flus. And this is why the prices of eggs were so high like 2 months ago. It's because there was tons of a bird flu outbreak in America, and we basically killed all the chickens on the farms in order to prevent that from spreading and turning into something that could potentially be highly pathogenic to humans. Okay, so we can get reassortment of the genetic components of a virus in order to produce new pathogenic forms. Or even if there's just a single strand of RNA, you can also get mutations within that strand that will change the surface protein sufficiently that it can evade our own immune responses. So this is the sort of mutational, the mutations that are accumulated here in the spike protein of COVID from the time of it was first sequenced and detected, okay, up until Omicron became the dominant variant, okay? And so they basically would sequence people that got sick, and this is the number of mutations that are different from the originating alpha strain. And over time, basically if you have a small number of mutations, okay, your antibodies against that particular shape of the spike protein are pretty effective, right? And so we had a big surge in cases, and then most people got the disease or vaccinated, okay, for that particular spike protein. Okay, and then the number of people that were infected with COVID dropped dramatically back down to, I don't know if you remember, but in June of 2020, it came right back down. We're like, oh, great, it's over. Let's open up everything. But then by the fall, things started to increase again. And then in around October of 2021, a totally sort of highly mutant form of COVID was able to reinfect the population, regardless of whether or not you'd been infected before, because it had over 25 mutations in the spike protein, which made it able to evade the immune responses of existing antibodies that your body had produced. Now, thankfully, it also made it slightly less pathogenic and less, well, sort of less deadly. Okay, so that also in some ways helps the fitness of that particular virus, because if you're killing your host, it makes it harder for you to spread. Yeah.
How successful like a virus is basically dependent on this like surface protein.
Well, that's the ability for it to evade the immune response is going to be dependent on the surface proteins, and the surface proteins will also help determine how easy it can its infectivity and what target tissues it can infect. Okay, so here's an example, right? So they developed all these antibody drugs, Paxlovid and different drugs that were designed to, bind on to, if you got sick with COVID, take the antibodies, okay, and the antibodies will neutralize. You take a large dose of them, and they're going to neutralize the virus that's replicating in your system, prevent it from spreading. Okay, here's the problem with that. Okay, those antibodies were designed for the alpha strain. Okay, so they were really good at getting alpha, the delta strain, this other variation. But then when Omicron came around, those antibodies useless. Did not bind to them at all. Okay, not every antibody was like that, though. Some antibodies did better than others, right? So there's sort of an arms race in a sense of developing antibodies that can continuously recognize new forms of the virus. Likewise, for the vaccines and mRNA, whether mRNA or other vaccines that are based on COVID, right? The vaccines from a single prior generation are not as effective as the actual generation of or variation of COVID that's circulating in the environment, because they're designed on the previous shape. There's usually some crossover, but not complete. Okay, so that's sort of the concept of antigenic variation. Now, what does the SARS target? Okay, so SARS, this spike protein on here targets what's known as the ACE2 receptor. ACE2 is involved in regulating blood pressure, okay, through this complicated process we'll talk about in the kidney unit. Okay. And the ACE2 receptor happens to be expressed on lung cells. So the spike protein will actually bind onto the ACE2 receptor and selectively infect lung cells. Okay, and that's where it's going to proliferate and grow initially. So that's why it's a very powerful respiratory virus, because if you breathe it in, the cells that it's targeting are right there in your alveoli. Okay. On the other hand, for the previous major, probably the other biggest virus of my life, HIV, human immunodeficiency virus, okay, that targets a different receptor, CD4. Has anybody heard of CD4 before?
Yes.
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 An hour ago.
Yeah.
Right. What did CD4 do? Where is that expressed?
Somewhere.
MHC2. It's expressed, it binds to MHC2, right? It's not, it's expressed on the helper T cells. And this is one of the reasons why HIV was so crazy insidious. Okay, there's two reasons. This is one of them. It selectively infected the helper T cells within your immune system. So it'll only bind to the CD4 cells and infect your helper T cells. That's one reason why it was really nasty. Okay, so when I was a kid, right, I will never forget, like 7th grade, sitting in class, and they announced Magic Johnson is retiring from the NBA because he has HIV. Okay, Freddie Mercury, Queen, Ryan White. I grew up in Indiana. This kid got a blood transfusion and developed HIV and AIDS. And so there was a sort of a huge explosion in HIV between 1980 and 1988, a huge explosion in awareness. A lot of money and funding went into trying to figure out how does the virus work and how can we develop treatments against it. And it's been actually incredibly powerful use and a demonstration that science can actually, it took a decade or two to really figure this out, but we have now developed very powerful tools, antiretroviral drugs that can really suppress HIV load to basically undetectable levels if you're infected. So what is the second thing that made HIV so insidious, so powerful, so dangerous? Okay, it is because HIV completely rewrote the book on how we understand molecular biology to work. Okay. The central dogma of molecular biology, as sort of promulgated by Francis Crick of Watson and Crick, discovered the structure of DNA along with Rosalind Franklin. He proposed that DNA gets turned into RNA, so DNA gets transcribed into RNA, RNA gets translated into proteins, right? And most viruses are made up of RNA. They just skip the DNA step. Okay, they just they package themselves up on RNA. RNA is less stable than DNA, but who cares? Viruses want to mutate anyway so that they can, evade the immune response. So they just go in and they inject your RNA into the body, and then you make your protein, and it's great. Okay, they're producing this. Okay, HIV does something very, very sneaky. It's made of RNA, but it can turn itself back into DNA, and that DNA can then get inserted into the genome, the stable part of your cells, and stay there for the entire life of that cell. Here's how it works. So we have our HIV particle. Okay, it has one of these coat proteins. It's like the spike protein on COVID, but instead it's called GP120. It binds selectively to our CD4 receptor. Okay, it is then going to insert its payload of RNA into the host cell, our helper T cell. Okay. Now, some of the proteins that are in this, one of the proteins, there's two really important proteins that are in this HIV payload. Okay, one of them is called reverse transcriptase. This is a protein that can take RNA and turn it back into DNA. So it takes the RNA, turns it back into DNA in the host. Okay. Then there's a second protein called integrase. This integrase can get into the nucleus. It travels with this DNA fragment of the virus into the nucleus, and it can cut the genome of the cell and insert its own DNA in there. So now you're getting a permanent installation of viral DNA into the genome of the host cell. So now the helper T cell is going to make RNA, even if you got rid of the viral particles here, it's still going to always have this the HIV DNA permanently embedded within its cells. Yeah. What? Not typically. You usually don't have, you can have recombination tools, but CRISPR is a little bit different conversation for another time. Okay. So then what happens, we're going to produce our protein molecules. Okay, those are going to get sort of packaged together, wrap up the RNA, and then you get a bud that comes out here, and then we're going to have a mature virion, which now is our HIV molecule that can then go off and look for other helper T cells to infect. Now, because it's integrated into this DNA, it doesn't always, the DNA can get turned on or turned off, depending on whether it's methylated, depending on how the chromatin's folded. Okay, so you can turn off this area of the gene, and it gets kind of inserts randomly into the genome. Okay, so it's in an area that's turned off and not being activated. It doesn't do much. It just sits there, waiting till that area of the genome gets turned on, and that might happen to some trigger a year from now. Then triggers that turning on of that part of the genome, and then it can start making the viral particles. So this is a concept called latency, where you have a delay between the acute infection and a reoccurrence of that infection later on. Okay, so for many viruses sort of can infect and then lay in a latent state. Herpes simplex virus is an example, right? Can be present in a host and come back during stress years later, over and over again. even if it's generally cleared by the immune system rapidly. And, basically the initial signs of this sort of epidemic of AIDS epidemic were really weird diseases. Kaposi sarcoma, which is like this cancer of the skin, yeast infections of the throat, okay, pneumonia, atrophy of the brain, really weird things that all had a similar common mechanism, which was the immune system had lost the ability to fight off a large variety of pathogens. Okay. And why did it do that? It's because the helper T cells got infected and slowly were getting killed off. This is what the overall trajectory of the HIV and AIDS progression looks like. Okay, so if you have, and this is after an untreated infection, right? We didn't have antiretrovirals are the class of drugs that block the that turning of the RNA back into the DNA. Okay. So very effective, but also very pretty specific to the HIV virus too, because HIV's approach is such a unique mechanism. Okay, so here in this plot, we have helper T cell concentrations in black. Okay. And then we have the concentration of HIV in the body and the concentration of anti-HIV antibodies in the body. Okay, so what happens first? Initial infection after a few weeks. We get a spike in HIV. Okay, HIV starts sort of infecting helper T cells. It starts producing viruses. The virus load goes way up. Then we start to make antibodies against it. Okay, we still have plenty of helper T cells around. They're not all infected. Okay, so the antibodies start going up. We have anti-HIV bodies that start going up. It starts killing off all the cells that are infected by HIV, neutralizes the HIV. HIV levels fall dramatically. Okay, but there's a secret pool of helper T cells that are infected by that DNA that haven't necessarily turned on yet. They're waiting for something else. Okay. And as each one of them turns on and starts producing those HIV particles, those particles are also displayed on MHC class 1. Cytotoxic T cells will come in and kill any helper T cell that's producing a lot of those particles, but that takes a process. And over time, the helper T cell population starts to get slowly killed off until at some point your helper T cell the number of helper T cells are so far down that you are no longer able to fight off other other pathogens, which is how that led to, this production of this appearance of all these other diseases that are the common symptoms of AIDS. Yeah. Right. So not also. Okay, so the helper T cell, right, is going to. Helper T cell is going to get infected by HIV. It's going to bind to the CD4 and get inserted. Not every helper T cell is going to get infected, right? Just, a subset of them is going to be sufficient to start producing HIV particles. But then the overall immune response, some other, B cells and helper T cells will recognize this particle and try to make antibodies and kill it off and suppress the virus. Okay. And then but that virus will have some latency. It'll be hiding in some set of a small number of helper T cells that are still have the DNA of the HIV in there, but in an area of the genome that hasn't been activated lately. Okay, so that DNA is not getting turned into RNA, not getting turned into protein until something happens to that cell, okay, that turns on that part of the genome, and now it starts to produce again. Okay. So part of the therapies that therapeutic strategies that people have tried to do is try to flush out try to activate all the helper T cells to identify areas of the genome that were capable of producing the HIV particle. But so far that approach hasn't been that successful, unfortunately. But the antiretroviral is pretty successful. Okay, any other questions about that? Could you repeat that?
If the red line doesn't.
Oh, so the red line doesn't drop over years. Well, okay, so those antibodies are going to be around there and they continue to circulate, okay? Because there's some pool of helper T cells and memory T cells and stuff that recognize that, and B cells that recognize the HIV, right? So the B cells are still there. There's still every so often, there's production of this HIV, these HIV molecules from a T cell that gets turned on to produce it. Okay, and so the antibodies are constantly at a high level. Okay, then because there's always there's always some helper T cell that gets activated. And when that helper T cell gets activated, starts making those HIV molecules, it gets killed very quickly. Okay, because the antibodies bind to it, target it for destruction. That cell gets killed. Yeah.
Is that helper T cell?
So that's a great question. So if the helper T cell gets infected by the virus, is the helper T cell going to make clone copies of itself? I think that would only happen in probably the rare case in which that helper T cell recognized the HIV virus with its T cell receptor, which most helper T cells are not going to recognize that virus. Only a small fraction of them will actually recognize the virus because they all have different T cell receptors. It would only replicate, it will only create copies of itself if it's a T cell receptor specifically recognizes the HIV virus. And the vast majority of helper T cells will not specifically recognize that virus because they have a diversity of different receptors or diversity of different binding. Okay, so you'll get a proliferation only of helper T cells that specifically recognize that virus. Regardless, so it's probably independent of whether or not that cell is infected or not by HIV. Yeah. Let's say suddenly there was some like super stressful event in your life, or you had some trauma to some area that turned on like the expression of some gene pattern there, or maybe there's a different infection that came in and activated a set of helper T cells that then somehow unveils that the activity within that part of the genome. Yeah. They sit there and so there'll be a subset of it. Okay, sometimes the genome, the HIV genome will insert itself, or the HIV DNA will insert itself into part of the genome that's turned on. Okay, and there it's going to immediately start making viral particles. Okay, with this stuff. But other times it's going to get inserted in part of the genome that's not turned on at the current time. Okay, so then if it gets turned on later, that's when that provides that reserve pool. Okay. So one last slide here. The antibodies as tools. Okay, so we can actually use antibodies. There's many different tools that we can use. We can harness antibodies through things like CAR T cells to target cancer and kill cancers within our bodies now through engineering antibodies. We can also use them as diagnostic tools for the detection of many different things. Okay, most notably things like, am I sick with COVID? How does that test work? Or am I pregnant? Okay, how does that test work? I'm not. But if I did get this, it would mean I have testicular cancer. Okay, if you test positive as a man on that, so how does this work? How does the pregnancy test work? Okay, immediately upon implantation of a fertilized zygote into the uterus, you get a sudden burst of human chorionic gonadotropin, a hormone that's produced. Okay, normally levels of that are extremely low, but then they go up by 1000x or more, which then sort of allows the uterus to start to produce the placenta. And so if we have grown up antibodies specifically that bind HCG, this particular hormone, we can embed them within this sort of test here, P on the strip. Okay, it's going to move the material. You'll have that vehicle of urine traveling through here, and if human chorionic gonadotropin binds to these immobilized antibodies on this strip, it'll cause a binding interaction that then causes a color change and allows you to specifically detect just that particular molecule, which is a unique signature of pregnancy and the fastest signature of pregnancy change within the body. So that is a way that we can utilize the incredible affinity and specificity of antibodies to make very specific tests for pregnancy, for viruses, for anything we want. Yeah. Human chorionic gonadotropin. We'll talk about that during sex and reproduction. All right, that's it. 10:50. Next week, kidney.